Hello students, welcome to the part 8 of Biomolecules chapter. In the previous classes, we discussed about carbohydrate, proteins as well as vitamin biomolecule. Today, we'll continue our discussion with one more important biomolecule, that is nucleic acids. You know, the particles which are present in the nucleus of the cell which are responsible for heredity. That means the transmission of inherent characters from parents to offsprings. Those particles present in the nucleus are called as chromosomes. And these chromosomes are made up of proteins as well as another type of biomolecule. So that biomolecule is nucleic acids. That means chromosome is made up of proteins and nucleic acids. There are two types of nucleic acids. One is deoxyribonucleic acid. In short, we call it as DNA. And another one is ribonucleic acid. In short, we call it as RNA. Okay, so regarding these two nucleic acids, DNA and RNA, you are learning in biology in detail, but in this chemistry chapter, chemical aspect of these two nucleic acids have been mentioned. That means today we'll discuss about chemical aspects of DNA as well as RNA. Chemical composition of nucleic acids. Nucleic acids are composed of three chemicals. First one is pentose sugar. Carbohydrate is there. A monosaccharide composed of 5 carbon atoms called as a pentose sugar. The second component of nucleic acid is we have a phosphoric acid moiety, and the third component is nitrogenous bases. Okay, different nitrogenous bases are there in a nucleic acid, and these nitrogen containing heterocyclic compounds are called as bases. These are the three components of nucleic acid: pentose, sugar, phosphoric acid, and nitrogen containing heterocyclic bases called compounds called as bases. Okay, well, we'll see these components. Pentose sugar. The nucleic acid RNA is composed of a beta D ribose sugar, pentose sugar, whereas DNA is composed of beta D2 deoxyribose sugar. And these two sugars, pentose sugars, are monosaccharides. Okay, so what's the difference between these two sugar units present in a DNA and RNA? DNA, the name suggests. So both are beta D ribose. Here also beta D ribose. The difference is that beta T2 deoxyribose. So difference is that in a RNA pentose sugar, H and OH are there, whereas at the second position, whereas in a DNA, H and H. So there is a lacking of one oxygen at the carbon number 2. Can you see? So here we have OH, whereas here at the carbon number 2 we have H, one of the position, isn't it? Therefore, it is beta D 2-deoxy here. 2-deoxy represents what? From the 2 carbon atom, one oxygen is removed from a beta D ribose. That's called as beta D 2-deoxyribose. And the bases, nitrogen base heterocyclic compounds, that means bases present in RNA are adenine, guanine, cytosine and uracil, whereas DNA composed of adenine, guanine, cytosine, whereas the fourth one is different, that's a thymine. So when you compare the bases present in RNA and DNA, the first three bases are same: adenine, guanine, cytosine. Here also adenine, guanine, cytosine, whereas the fourth nitrogen base is different. So here in case of RNA we have uracil, whereas in case of DNA we have thymine nitrogenous bases. So these are the two components, pentose sugar as well as a nitrogenous bases. Apart from these two, the two nucleic acid contain phosphoric acid moiety. Okay, we'll move to the next concept. We'll see structure of nucleic acid. So in the beginning itself I said you that we are learning structural aspects of nucleic acid. So how is the nucleic acid? Actually the nucleic acid is a polymer. It's a polynucleotide. So how that polynucleotide is formed, we'll see now. First of all, a unit formed by the attachment of a base, a nitrogenous base, to one dash position of a ribose sugar is known as nucleoside. That means first we'll see what is meant by nucleoside. Nucleoside andre ಏನು ಹಾಗಾದ್ರೆ? So when a base gets attached to ribose sugar, so ribose sugar ಗೆ base attach ಆಯ್ತಂದ್ರೆ ಯಾವ position ದಲ್ಲಿ? First carbon. So here the carbon numbers in a nucleic acid sugars are numbered as a 1 dash, 2 dash, 3 dash and so on. So we'll see the structure. So what's the nucleoside then? Base attaches to the ribose sugar. Okay. See here in this slide. So base react, that means gets attached to the ribose sugar. I have taken a RNA sugar, beta D ribose. So what is formed? A nucleoside is formed. This is the structure of a nucleoside. So can you see? So a pentose sugar is there to this 1 dash position. So I said the carbon atoms in a ribose sugars are numbered as 1 dash, 2 dash, 3 dash, 4 dash and 5 dash. In case of carbohydrate we used to number them as a 1, 2, 3, 4 and so on, isn't it? Okay, now this base gets attached to 1 dash position. Now this structure is called as what? A nucleoside. Clear? Very simple. It's a unit formed by the attachment of a base to 1 dash position of a ribose sugar is called as what? Nucleoside. Okay. When a nucleoside is linked to phosphoric acid moiety, at which position? At a 5 dash position of a sugar moiety is called as a nucleotide. So now we'll see how a nucleotide is formed. To the nucleoside, look here, to the nucleoside, if a phosphoric acid moiety gets attached, at which position? At a 5 dash position, we'll get a nucleotide. So structure we'll see. So this is a nucleoside formed in the first step, combines with, gets attached to H3PO4 phosphoric acid moiety. At a 5 dash position we are getting this structure. So this structure is called as what? A nucleotide. So nucleotide it is. So ಇಲ್ಲಿ ಏನಾಗಿದೆ ನೋಡಿ ಹಾಗಾದ್ರೆ, this is the sugar unit. Okay, to that base is attached, called as a nucleoside. To that nucleoside what is attached here? A phosphoric acid moiety is attached. Okay, so in the structure bonds are not visible here. Between carbon and oxygen bond is there, between phosphorus and oxygen bond is there, here also bond is there, bonds are not visible. Hagadre this unit is called as what? It is a nucleotide. Clear? Okay. These nucleotides are joined together by a link, a bond called as a phosphodiester linkage between 5 dash position and 3 dash position of a carbon atom of the pentose sugar. So in a previous step what we discussed, a nucleotide is formed from a nucleoside and a phosphoric acid moiety, isn't it? So these nucleotides, 1 or 2 or more nucleotides, are joined together by a phosphodiester linkage. Okay, look at the structure. So this structure is what? There is a sugar, to that base is attached, to that phosphoric acid moiety is attached. Hagadre this structure is called as what? Yes, it is a nucleotide. One nucleotide joined together with another nucleotide. So can you see here? So in this slide you can see one nucleotide, another nucleotide. These two nucleotides are joined together. ಯಾವ position ದಲ್ಲಿ ನೋಡಿ? 5' position of one nucleotide with the 3' position of another nucleotide. ಹಾಗಾದ್ರೆ ಸೊ ನೋಡಿ, this is 1', 2', 3'. ಹೌದಾ? ಇಲ್ಲಿ here, okay, we'll see the diagram now where exactly the link forms. Look here, this is 1', 2', 3'. So 3' moiety of one nucleotide gets links with another nucleotide. Where exactly the linkage is seen? Can you see here? 3' gets linked to 5' through a link, through a bond. That bond is called as what? That bond is called as phosphodiester linkage. Okay, this linkage is called as what? Phosphodiester linkage. So a unit formed by the attachment of two nucleotides is called as what? It is called as what? It is called as a dinucleotide. Okay, suppose to the dinucleotide one more nucleotide gets attached, trinucleotide. To that one more, tetranucleotide. Likewise, when many nucleotides are joined together by a phosphodiester linkage, we call it as what? Polynucleotide. Isn't it? Okay, so this linkage, no, in the previous biomolecule, that's a carbohydrate biomolecule, we come across one linkage between the monosaccharide unit, that's called as what? Glycosidic linkage. Can you remember? So in proteins we come across peptide linkage, whereas in nucleic acid we are come across one more linkage, one more bond. That bond is called as what? Phosphodiester linkage. Okay, so clear how a dinucleotide is formed? When two nucleotides are joined together at a 5' and 3' position through a phosphodiester linkage, we are getting dinucleotide. Very simplified version of a nucleic acid chain we'll see. So what is nucleic acid? Nucleic acid is a polynucleotide. So look at this simplified version. We had a sugar. To the sugar, if a base gets attached at a 1' position, sugar ge base attach ಆಯ್ತಂದ್ರೆ ಏನಂತಾರೆ ಅದಕ್ಕೆ? ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್. ಆ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ಗೆ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್ ಅಟ್ಯಾಚ್ ಆಯ್ತಂದ್ರೆ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಂತಾರೆ. ಆ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಮತ್ತೊಂದು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಗೆ ಅಟ್ಯಾಚ್ ಆಯ್ತಂದ್ರೆ ಡೈನ್ಯೂಕ್ಲಿಯೋಟೈಡ್, ಮತ್ತೊಂದು ಟ್ರೈನ್ಯೂಕ್ಲಿಯೋಟೈಡ್. Likewise, if n number, can you see here number n? So if n number of nucleotides are joined together by n minus 1 phosphodiester linkage, we are getting this kind of a chain. Sugar base phosphate, sugar base phosphate, sugar base phosphate. Likewise continues. Okay, so this is a polymer chain called as what? Polynucleotide. Polynucleotide is nothing but nucleic acid. Clear? Hagadre DNA and RNA are composed of what? This kind of a polynucleotide chain irtade. Clear? Okay. And this sequence of nucleotides, arrangement of nucleotides in the chain. Can you see in this sequence? So this sequence of arrangement of nucleotides in a chain is called as what? Primary structure. We come across primary structure in a protein, no? Isn't it? So likewise here also we have a primary structure, the sequential arrangement of nucleotides. In a nucleic acid we come across primary structure as well as a secondary structure. Whenever a secondary structure is formed, when a primary structure gets folded, no, we are getting a secondary structure. We have discussed in a protein biomolecule. The same thing we come across here also, when the nucleic acids also have a secondary structure. Very good example is Watson and Crick model of double helical structure of DNA. You are learning in biology. Okay, James Watson and Francis Crick gave a double strand helix of structure for a DNA. That structure is what? A secondary structure. One more important aspect in this polymer chain is that, in this polynucleotide chain, is that the two nucleotide chains are wound about each other and they are held together by a bond. It's a hydrogen bond. So where exactly the hydrogen bond is present? It is present between the pairs of a bases. Okay, so the two nucleotide chains are held together by a bond, hydrogen bond. So hydrogen bond is not present between sugar unit. No, it's not present between phosphate group. It is present between bases, adenine, guanine, cytosine, thymine and uracil bases we can no. So between those bases nucleic acid, what you call hydrogen bond is there. Because of this complementary base pairing, the two strands looks alike. The two strands of nucleotide chains are complementary to one another. They looks alike. Why? Because the hydrogen bond formed are between a specific pairs of bases. We'll see in the next slide how exactly the specific base pairing is present. Always adenine pairs with thymine. So hydrogen bond adenine base ಮತ್ತು thymine base ನಡುವೆ ಅಷ್ಟೇ ಇರ್ತದೆ. Adenine will not form a bond with a guanine. Adenine will not form a bond with a cytosine. So adenine always form a hydrogen bonds with thymine and guanine always form a bond with cytosine. So this is specific base pairing. Adenine will not form cytosine bond, guanine will not form thymine bond, adenine will not form a guanine bond, thymine will not form a cytosine bond. So this adenine, thymine and guanine cytosine specific base pairing is there. That's why the two strands of a nucleic acid in case of DNA are complementary to one another. ಅಂದ್ರೆ they looks alike. ನೋಡ್ಲಿಕ್ಕೆ ಒಂದೇ ತರ ಕಾಣಿಸ್ತವೆ. In case of RNA, the second structure of RNA, so helices are present, but they are not double stranded. In case of DNA, it's a double stranded, whereas in case of RNA, it is only single stranded. So can you see in this diagram? So it's a DNA structure. This is RNA structure. So can you see this red colored dotted structure? This is one polynucleotide chain. So this blue colored one, another polynucleotide chain. Okay, so two polynucleotide chains are wound about each other. So can you see these lines, white color lining? Can you see? This is what? Hydrogen bond between bases, adenine, thymine and guanine as well as a cytosine. Whereas in case of RNA, we seen only a single strand. Okay, secondary structure is single stranded. There are three types of RNA molecules and they perform different functions. So the three types are mRNA, rRNA, tRNA. mRNA is called messenger RNA, rRNA is called ribosomal RNA and tRNA is called transfer RNA. So regarding these three types you are learning in detail in biology. Okay, so ಇಲ್ಲಿ ನಾವು ಕಾನ್ಸಂಟ್ರೇಟ್ ಮಾಡ್ತಾ ಇರೋದೇನು ಬರೀ ಸ್ಟ್ರಕ್ಚರ್ ಬಗ್ಗೆ, ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಹೆಂಗೆ ಫಾರ್ಮ್ ಆಯಿತು, ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಹೆಂಗೆ ಫಾರ್ಮ್ ಆಯಿತು ಅದ್ರ ಬಗ್ಗೆ ನಾವು ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾ ಇದ್ದೇವೆ. ಫೈನ್, ವಿಲ್ ಮೂವ್ ಟು ದ ನೆಕ್ಸ್ಟ್ ಪಾಯಿಂಟ್. Biological functions of nucleic acid. Okay. DNA, already I have told you in the introductory part, it's a chemical basis of heredity and it's called as a reserve of genetic information. So the genetic information is present in a DNA nucleic acid. And this DNA molecule, it's capable of self-duplication during cell division and identical strands are formed and they are transferred into the daughter cells. Another important function of nucleic acid is protein synthesis. Actually, the protein synthesized by RNA molecules, various RNA molecules in the cell, but the message for the synthesis of a particular protein is present in a DNA. Okay, so these are some biological functions of nucleic acids. Okay, so very short concept we have in our syllabus. So in study ಮಾಡಿದ್ವಿ ನಾವು nucleic acid structure, a nucleoside formation, nucleotide formation, phosphodiester linkage, isn't it? Then sugar units present in a DNA as well as in a RNA, then a phosphodiester linkage, specific base pairing, and 4 different types of nitrogenous bases, isn't it? So based on this concept, you will have 1 mark question. So previous question paper nodidre 1 mark prashne bartade, and the question will be very simple. We'll see which all questions have been asked in previous year annual examination question paper. Okay, one by one we'll see. March 2014 question. 10th question was like this. Which is the nitrogenous base present only in RNA but not in DNA? So very simple question will be there, no, for 1 mark. So it will be assignment for you. July 2014, which is the nitrogenous base present in RNA but not in DNA? Same question. March 2015, name the base present only in DNA but not in RNA. In July 2015, which is the nitrogenous base present only in DNA but not in RNA? Same question. July 2016, 36th B question was like this. What is a nucleoside? For 1 mark. So ಹಾಗಾದ್ರೆ ಪ್ರತಿ ಕ್ವಶನ್ ಪೇಪರ್ದಲ್ಲಿ ಎಷ್ಟು ಮಾರ್ಕ್ ಕ್ವಶನ್ ಬಂದಿದೆ? 1 mark question we can expect. In the next March 2017 question paper, name the nucleic acid responsible for genetic information. 1 mark question was there. July 2017, name the pentose sugar present in RNA molecule. March 2018, which is the nitrogenous base present only in RNA but not in DNA. July 2018, name the nitrogenous base present only in DNA but not in RNA. July 2016, 36th C question was like this. Name the sugar moiety present in DNA. Okay, so ಇವೆಲ್ಲ ಕ್ವಶನ್ ಪೇಪರ್ಸ್ದು ಕ್ವಶನ್ ನೋಡಿದರೆ ಏನು ಕನ್ಕ್ಲೂಡ್ ಮಾಡಬಹುದು? You may get 1 mark question from this nucleic acid concept. Clear? Base, sugar units, then nucleoside, nucleotide, then biological importance, specific base pairing. You remember this concept, that is more than sufficient for your annual examination point is concerned. Clear? Okay, so we'll see one animated explanation or animated video on nucleic acids now. Okay.
Nucleic acids are a class of biomolecules that are found in all living organisms and are responsible for the storage of genetic information. They are also responsible for protein synthesis. Nucleic acids are made up of monomers called nucleotides. Nucleotides are made up of 3 components: a 5-carbon sugar, such as ribose, a phosphate functional group, and a nitrogenous base. The 5-carbon sugar present in DNA is deoxyribose, and the 4 nitrogenous bases are adenine, guanine, cytosine, and thymine. Nucleotides are linked together by dehydration synthesis, or polymerization reactions, between the sugar of one nucleotide and the phosphate group of a second nucleotide. In the process, a sugar-phosphate backbone is formed, linking the nucleotides together. Notice how the nitrogenous bases project to one side of the backbone. DNA is composed of 2 strands arranged as a right-handed helix. The strands are also anti-parallel, meaning that they are oriented in opposite directions. The 2 strands are held together by hydrogen bonds. Guanine forms hydrogen bonds with cytosine, and adenine bonds with thymine. This complementary base pairing links the strands together. It also plays an important role in the replication of the DNA molecule. RNA differs from DNA in that it contains the sugar ribose instead of deoxyribose and the nitrogenous base uracil instead of thymine. Also, RNA is usually single-stranded and does not typically form a helix. Because of this, it is less stable than DNA and is usually more temporary. While DNA stores the information that is needed to construct a protein, RNA functions in regulating the expression of this information in the protein synthesis process. Not all nucleic acids are involved in information processing in the cell. ATP.
Okay, so this is about today's concept, study of nucleic acid biomolecule. Okay, so far we have completed the study of carbohydrate biomolecule, the study of protein biomolecule, vitamin biomolecule, as well as nucleic acid biomolecule. A very short concept is left in this chapter, that is study of hormones, that we'll discuss in the next class. Thank you very much.